Okay, thank you. So I'll be sharing my slides. I hope you can see them well. I have quite a provocative title for my presentation. I'm now opening my chat to also follow the questions that we are receiving. So as I said, it's a little bit provocative. How to study cognitive disorders? Well, we'll probably mostly be talking about the way we are studying the cognitive disorders. And we'll be talking about new opportunities that have emerged recently in Russia. Maybe not everybody is aware of these new opportunities. If we talk about cognitive disorders, we can be quite different here. So Philip Haitovich will be talking about molecular and biochemical level, but I'll be talking about neuroimage. So this is the level at which we use different methods, fMRI and various ones, and it allows us to study different processes in our brain. Those are not invasive methods. So I'll be describing different approaches towards our cognitive health. I'll be explaining our logic, and I'll also be talking about what's new is there in the Russian Federation. And it will allow you maybe to use some new opportunities while studying cognitive functions and cognitive health. So proceeding from our previous discussion, I have changed my presentation a little bit. The cognitive level, as has already been said, is quite a mysterious one. We do not know exactly what is a cognitive, how actually the brain activities are translated into cognitive processes. We are not fully aware of these biochemical processes and how they're transferred into decision-making. And this is quite well demonstrated while we perform some classical interventions, when we help people with different cognitive disorders. So I took a couple of questions from an overview on the classical methods of aid and help, and they're connected to the administration of different medications. So here we describe different processes. For example, we give a medication, it helps, but what has been enhanced by drugs? Which process has been enhanced, which process has been suppressed? And without understanding cognitive processes, it will be quite difficult to understand and to expect some particular results of the intervention. So you can enhance one function and then offset the other one. What is the efficiency of what is going on? It should be assessed on a cognitive level. And as I've already said during our discussion, one of the leading medications that helps with the Alzheimer's disease, well, they have the 2 points on the whole scale. So my idea here is that it helps, but we should understand the consequences of an intervention. Are there any consequences? Well, usually there are some. And who from your participants of the experiment reacted to this intervention and who didn't? And here we face the individual consequences for a particular person from a medication. They could be different consequences, not only proceeding from the pharmacological interventions, but also proceeding from the behavioral ones. And so this is the focus of a study of intervention when we help people with cognitive disorders. Well, neurocognitive disorders can be determined quite differently. We have a lot of schools here, according to the number of the disorders. But here we see that those are connected to the all basic cognitive functions that can be found in any course book. Those are disorders of motor functions, attention, cognitive control, memory, training, language, and social cognition. And here we will go back to our panel discussion again. Those are very different levels when it comes to their complexity. Some levels can be attributed to some local processes that are occurring in some brain areas, but some of them are more social. So here we should study not only one brain, but we should be examining the interaction between different brain systems. So the approaches towards the study of cognitive disorders can be quite different, and we have chosen one for ourselves. I'll be referring to that today, and I'll be talking about our Institute of Cognitive Neurosciences and about our partners at HSC. So definitely we understand that there are different trends in the research of cognitive disorders. And Konstantin Anorkin was also mentioning some very important and interesting neuroprocessing and interaction between different neurons at the same time. So here, I probably agree with the opinion of the doctors that say that one of the main areas of this research are the biomarkers to diagnose, then the early prevention of neurodegeneration, and the next one is about the monitoring of the treatment and monitoring of the efficiency of the methods available. That's the way we also try to proceed with. And this is an overview of the neuroimaging-based markers in psychiatry. Here we can see MRI, which is dominating the CAT studies as well, but a lot of biomarkers are also connected to the structural changes in our brain. Well, this is a very good and understandable classics, so to say. If we see some structural changes happening, then we can also forecast some disorders. In our labs, we took a little bit different approach. If I could use the graph of my colleagues, then I need to say that our understanding of a disorder, is more dynamic. So we see the brain work as not a work of one area, but as a very comprehensive and complex network of different areas. And this is the dominant approach in the cognitive biology today. So we should not be talking about just one neuro area. If we want to understand the symptom, but not only the symptom, but also the reason, the cause of the disease, then we should understand what was the root cause of that and what results in particular symptoms. So that's why we focus on the work of the distributed neural networks. And proceeding from that rules and laws, we try to identify the persistent markers in our brain that actually determine our behavior and our cognitive functions across our whole life. Those are some dynamic processes that can be changing. They can be quite fast in changing because of an intervention or because of a disease. And this altogether allows us to develop our methods and approaches and to create some new methods of neuromodeling of the brain activities, and they are quite adaptive to the states of your brain. This could be adapted to some individual peculiarities or some dynamic peculiarities as well. And here we take a look at multimodal approaches. As the practice shows, we cannot create markers efficiently when we take only one approach. And here I need to mention Vadim Nikulin. He's already been speaking in here, and we have some joint research in that respect. And he's been developing the idea of some dynamic characteristics of neural nets. If you missed his presentation, I strongly advise to follow his presentation. It's very interesting. So he develops the idea of some mathematical laws. And this is about the idea that there is a balanced state of a brain. This is optimal for receiving information, for our behavior, for providing for the cognitive functions. So you can have some subcritical state. If you have this pile of sand, then you can have a critical slope. This is a high hill, and this is very sensitive to each of its elements. So if some small elements will be added, then you'll be facing an avalanche. So this is a very sensitive system, and it can be sliding into some unstable state at all. And so this analogy is a very interesting one. It's done by Vadim, as I said. And so he focuses on this balanced state of a brain and of the brain functions. And here we see also very interesting research. We've been doing it together with our German partners. And here we show the state of our brain, and it can also be a marker of some particular interventions. So here we can see the long-standing correlations that Vadim has been mentioning, and this is about the criticality of the balanced state of the brain. So we can see these markers before an intervention and after that. And here we can see the changes. And on the right, you can see quite clear correlation of these markers with the changing symptoms. So the characteristic of these local processes can also be a very efficient marker of what is going on. What is the most efficient medication? We got this question. Well, I'm not a doctor, so I think that we should refer these questions to doctors or to Philip, who's been dealing with this kind of issues. So I'm a neurobiologist, and I'm focusing on cognitive activities. Another work, which is about to be published in Stroke journal. This is by Marina Nazareva, and she's a doctor and she's a scientist at our center as well. And she tried to combine parameters that will forecast the restoration after a stroke. And she focused on two modalities here. First, on the connections in our brain. And she got it with the DTI and MRI. And also she studied the reaction of a patient's brain to some kind of activities. And so this shows that those methods can forecast quite clearly for different groups of patients. But if you really want to divide patients across 3 groups, for example, then you would require a combination of these methods that would result in 100% of the clearness and precision of the results of the restoration after a stroke. And this kind of analysis of a complex and comprehensive neural dynamics in online MRI also have been used by us. I'll not go into the details here. I just would like to remind you that we are focusing on the two-directional interface here. So here we have neuronal recordings. We've been reading the information from a monkey's brain, and then we also translated it into the avatar work, and then we got it back to the brain. And so this is a multimodal project. Here we're reading the activities of the brain, and we also stimulate the brain. And this multimodal system is also quite fitting for Michael's projects, and I believe that he'll be developing in the same direction at Skoltech. So our approach boils down to the fact that the cognitive health is connected to the distributed systems in our brain activities. And if we can assess it, and if we can assess the dynamics of this system's work, then we have some persistent characteristics of these dynamics, and we have also fast-changing characteristics. And by this, we can create the methods of marking the cognitive state of a person. And those markers would be individualized. They would allow to apply brain stimulation methods and adapt them to some particular brain condition and state. So that's how and why we use multimodal approach. And these interventions can be not only performed in the form of an intervention or medication, we can also go to some other interventions. And here I'd like to touch upon the topic that has already been mentioned. I would like to talk about the changes that will occur to us in any ways with the age. So after 45 years, our cognitive functions start to deteriorate. And the majority of us, if we live up to 100 years, will face this or that cognitive problem in any case. And so the problem which we've been developing in the cognitive neurobiology is that across our life, these are our cognitive resources of our brain. They are developing, but then starting from some particular age point, they start to deteriorate. And the worst piece of news here is not that they're just deteriorating, but that they're deteriorating at a very risky state. And here we start to talk about the intervention. We can enhance our cognitive resources at some point of time. And it goes quite well with the idea of Philip that has already been mentioned. So we can forecast for the future to accumulate some additional cognitive reserves for our aging future. So here we can enhance these resources to such a level that when we go down afterwards, indeed we will face some problems, but they will not be critical. So we will not reach the level when we will be feeling impaired or we will experience some cognitive disorder. And so here we work across different approaches. Here you can see Matteo Fiura, a significant scientist. On the one hand, he develops the individualized approaches to the brain stimulation. So he uses these or those biomarkers and he tries to choose the individualized protocols of the cognitive functions influence. And we have another approach here, neuroenhancement. This is about the idea that some cognitive functions can be accelerated a little bit more, even much more. And here we also talk about alternative current neurostimulation. When we use some particular length of the current and we apply to a particular area of our brain, then you can really enhance some functions of our brain. And this approach should be very much individualized. Another interesting area of development here is about using the markers, multimodal markers, of a cognitive state. And this is done in our group that is studying speech. And this is the result. Here you can see across the vertical axis, you can see the results while people are performing some difficult tasks. And the x-axis is about the volume of the gray matter. And here we have different colors, and they indicate the level of a second language fluency. And so here we can see the proportion. Depending on the level of your knowledge of the second language, you can also track the neuroactivity in your brain. So it's very interesting because without any invasion, you can actually model some functions. With the study of the second language and the level of this language fluency at a person, you can also monitor the cognitive resources and you can enhance them. This is the line that we have been using in our studies. Take it comprehensively. We believe that we can study dynamic and polystatic markers of these neural networks. And we can come up with methods that are adaptive to our brain and to our particular states. We do use different approaches. And here, Konstantin Anokhin has already shown this classical scheme from the course books on neurobiology. Here you can see different methods. Across the vertical axis, we have the spatial dimension, and across the x-axis, we have the time dimension. So here we can see different methods. Sometimes some of them are quite well localized in the brain area. Some of them are quite well localized across the space, but I should indicate that there is no ideal method. Which methods should we use during our cognitive research and interventions? This is a very good question. If we pick out a method, then what methods are available in Moscow? EEG, MRI, MEG. So they are quite similar when it comes to the spatial dimension. Some of them do have some advantages, but they're different when it comes to the timeline. So MEG and EEG can study neurobiological correlations quite well, but when we talk about MRI, then it'll be quite slowly done. But if we take a look at the price, the selection, and in the invasive factor, here we can see the difference. So there is no ideal methods, and that's why I say that we should have them in combination. So we do have very good centers of expertise in neurobiology and cognitive neurobiology in Russia. Those are located in St. Petersburg, in Moscow, in Kazan, in Novosibirsk. Lately, we've come up with some new participants in place that we should be aware of. So that was done due to the system of mega grants. We have two prominent labs in St. Petersburg by Yuri Shturov. They're studying speech processes, and also the lab by Yelena Grigorenko, and there they are studying aging processes and their connection to cognitive functions. There are some new projects also due to the mega grant. For example, in HSC, we also have such a project. In our lab, we also have a very interesting lab of psychogenetics and its connection to the age. In Skoltech, we also observe the development of cognitive developments and researches quite a lot. So this is the answer to the young scientists who ask, where could they apply their knowledge? And we also we received these questions during our discussion. How can we make the centers attractive for our youngsters? Well, they have been developing very fast, and we have to monitor these processes. And in our center, in our lab, we took a decision to equip our lab with some baseline methods. I believe that we have the leading system on the brain stimulation, from transcranial to the stimulation with the alternative or other type of current. We have EEG, etc. We also have a joint laboratory together with one of the institutes. This is on the MEG. We also establish relations with the clinical basis due to our partners and our project on invasive interfaces. We also have good relations with the MRI centers as well. But as Konstantin Anokhin has properly said, we have 2 or 3 MRI scanners in this country that actually can be focusing on research. So as of today, we have been quite well equipped. And here I'd like to mention another aspect of cognitive sciences and cognitive disorders, and it is connected to the cross-functional study. So, for example, here we can see the classical course book, and if we take a look at the chapters, we see everything here, from the cognition to emotions to language to the cognitive control, social cognition, and evolutionary perspectives. So we do understand that cognitive problems are quite interconnected. We can visualize it in this kind of a platform and pyramid. So we understand that we're proceeding from some kind of a molecular and biological level, then to a more complex one, to neurons, to the brain, to the cognition, to the social aspects, and to the environmental aspects. That's what we have been discussing today. So the genes, the biochemistry, the society, the evolution, everything influences us. It's understandable that historically we've been studying these areas across different neuro and cognitive sciences. What we have tried to do, we try to synthesize all these levels at HSC. Well, evidently, this is based on the mathematical modeling. I just wanted to demonstrate it with a couple of slides, the interaction between different levels. So the social level can be interacting with the biological one quite well. We have a lot of examples related to that. Those systems can be quite different. For example, the multiple determinism of different functions. So we have, for example, biological and social factors coming all together. For example, drug abuse and addiction on the biological level has been also determined by some genes, some specific receptors. But when it comes to the social level, drug abuse and drug addiction is also highly determined by the social environment where we live. And this influences our behavior and cognitive functions. For example, another sample, testosterone and the sexual behavior of primates. This could also be studied from different aspects. So we have biological aspects and the social aspect here. And another example, the third one here, it's very interesting for us as well, non-additive determinism. So the specifics of a whole, of the social whole, cannot be boiled down to some biological ones. So here we studied the monkeys, and we understood that the social level could be also be influencing the social structures and the behavior of the monkeys. So the cognitive processes are very interdisciplinary here. Here we can see the neurobiology, humanitarian sciences, computer sciences, etc. So our tradition has this flaw, which is characterized by the weak social and other aspects. And we believe, we hope that we will open our cognitive center in the almost very center on Moscow. This is the building that is planned. So we will have the transcranial magnetic stimulation, also near infrared spectrography. Then we will have the transcranial magnetic stimulation MagVenture. We will have a couple of stimulators, and they'll stimulate a couple of brain areas. We will also have robotized systems that will allow you to, or us to, conduct some very comprehensive and difficult trials. Then we will also have the cognitive floor. There we'll be studying cognitive functions development, the eye tracking, the 3D vision, and 2D vision as well. And on the next floor, we'll have the brain-computer interfaces lab, and also the neuropsychology lab. This is a mega grant received by Olga Dragoy. They're studying speech disorders. And on the fourth floor, we'll have the social neuroscience and neuroeconomics groups working. So we try to synthesize all the levels of cognitive processes, from the perceptive ones to the social ones. Going back to the question whether our research can be made attractive to the youngsters, well, we started our project at the eve of 2014, and it took us 5 years to reach quite a good level, to become visible, and to become quite attractive for our young scientists. And for those who are hearing about our Institute of Cognitive Sciences for the first time, I'd like to present our opportunities and capacities. 2 research labs. Also, we are specializing in neurodynamics, the neuropsychology of the speech, neuroenhancement, cognitive enhancement, the modeling of addictions, and also the impairments of decision-making. And by this slide, I'd like to finish my presentation. So we perceive cognitive processes as a comprehensive, difficult, spatial and time-split processes. And we use different methods of biomarkers and individualized methods of interventions. With this, I'd like to thank you for your attention, and you can always reach our website of our Institute of Cognitive Neurosciences, and thank you very much for this opportunity to be presenting at this conference.